On this second lecture, we will talk about metabolism of bacterial cells. First, we will stop on aerobic metabolism, or we can call it aerobic respiration. And then in the end of this lecture, we will compare metabolism of aerobic bacteria to metabolism of anaerobic bacteria. Metabolism is a chain of biochemical reactions that take place in a cell. Metabolism is complex, but at the same time, it is completely logical. It is step-by-step process by which cells take nutrients from their environment, convert them into cellular components. As a result, cells grow, and then they divide and produce two new cells. As soon as a new cell formed by cell division, it starts collecting nutrients from environment. And when the cell has doubled its mass, it divides again. The process continues as long as environment is favorable. The steps of this process are called biochemical reactions, and all they are catalyzed by enzymes. Metabolism of most living things is similar, but there are some differences depending on the kind of organism and condition under organism is growing. Metabolism of bacterial cells has 5 metabolic tasks. The first task is called bringing nutrients into the cell. Means cells start transport of nutrients across the plasma membrane, and then they concentrate them in cytoplasm. And the next task is catabolism. Catabolism means breaking down. So during this task, cells break larger molecules of nutrients into smaller organic compounds. Next task is biosynthesis. By biosynthesis, cells make building blocks. Those are small molecules that are very specific for each cell. And then polymerization links together building blocks and forms macromolecules. And the last metabolic task is assembly, means cells assemble macromolecules into cellular components. For example, cell walls, ribosomes, flagella, pili, and so on. This slide shows you that all metabolic tasks are connected to each other and depend on each other. Remember, the first metabolic task is called bringing nutrients into the cell. And you see how nutrients go across the cell envelope. As soon as they get into cytoplasm, now we call them substrates. Next, substrates will go through catabolic reactions. Remember, catabolism means breaking down, means those substrates will be broken into smaller molecules. We call them precursor metabolites. You see, a cell will form 12 precursor metabolites. That's right, cell requires only 12 precursor metabolites. Besides precursor metabolites, ATP molecules will be formed and reducing power components. Next task is called biosynthesis. So precursor metabolites will go through biosynthesis, and remember I told you biosynthesis forms building blocks, or we call them monomers. Those are small molecules specific for each cell. Building blocks then go through polymerization. When they go through polymerization, they will be linked to each other. Building blocks will be linked to each other and form macromolecules or polymers. And then the last task is assembly. This is when polymers will go through assembly and form cellular components. Alright, so the first metabolic task is called bringing nutrients into the cell. It means nutrients have to go from environment into cytoplasm across the cell envelope. Remember, on the previous lecture I told you that the cell envelope of gram-positive bacteria is composed of two layers: plasma membrane and cell wall. Cell envelope of gram-negative bacteria is composed of three layers: plasma membrane, cell wall, and outer membrane. And since cell envelope of gram-negative bacteria has three layers, we are going to talk about first metabolic tasks on an example of cell envelope of gram-negative bacteria. On this picture, you see a picture of the cell envelope of gram-negative bacterium. So the most inner layer is called cytoplasmic membrane or plasma membrane. And then the next layer is called cell wall. And then we have outer membrane, the most outer layer. Let me remind you that the space between plasma membrane and outer membrane is called periplasm. Since outer membrane is the most outer layer of a cell envelope, it is the first barrier for nutrients. So how nutrient molecules go across outer membrane? They use the most common method or the most common way. We call it simple diffusion. So what is it, simple diffusion? Simple diffusion always follows concentration gradient. That means concentration of those nutrient molecules outside of outer membrane is much higher than concentration of the same molecules inside of periplasm. Alright, now nutrients are in periplasm. So the next layer of cell envelope called cell wall. Remember, cell walls of all bacteria are made of peptidoglycan. And remember from the previous lecture, peptidoglycan is a mesh-like structure. So that means it is not a barrier for nutrients. They go across the cell wall without any problems. But plasma membrane or cytoplasmic membrane will be the last barrier for nutrients before they get into cytoplasm. Remember, plasma membrane is made of phospholipids, means lipid-soluble molecules can go across a plasma membrane without any problems. But most nutrients are not lipid-soluble molecules. Then how do they get into cytoplasm from periplasm? They have to go through holes within plasma membrane. We call them pores. And remember, I told you those pores are made of proteins. We call them transporters. There are two ways for nutrients to go through pores of the cytoplasmic membrane. They are facilitated diffusion and active transport. Let's talk about facilitated diffusion first. The main characteristics of facilitated diffusion are: it always follows concentration gradient. means concentration of nutrients in periplasm must be higher than concentration of the same nutrients in cytoplasm. And the second characteristic, it requires transporters. Those are proteins or enzymes. They have ability to recognize certain specific molecules in periplasm, attach to them, and then bring them across cytoplasmic membrane into cytoplasm. Active transport also has two characteristics. First characteristic, it requires transporters or enzymes. And the second characteristic, it goes against concentration gradient. It means concentration of those nutrients will be in cytoplasm higher than in periplasm. So basically, active transport has to pump those nutrients into cytoplasm. So because it works as a pump, like a pump, it requires energy. The next metabolic task is catabolism. And catabolism will break down a larger molecules of nutrients into smaller compounds. We call them precursor metabolites. And I told you cells require only 12 precursor metabolites, and by the way, human cells also form only 12 precursor metabolites. This slide shows you that all 12 precursor metabolites will be formed by 3 catabolic pathways. Those are glycolysis, TCA cycle or tricarboxylic acid cycle or Krebs cycle, and the last one is pentose phosphate pathway. So the first 6 metabolites will be formed by glycolysis. The next 4 will be made by TCA cycle, and the last 2 by pentose phosphate pathway. Besides 12 precursor metabolites, catabolism makes ATP molecules or adenosine triphosphate molecules, which are a source of energy for cells. And also catabolism makes reducing power compounds, compounds that participate in many biochemical reactions. This picture shows you that in metabolism everything is connected. It's like a cycle. For example, when complex or large molecules go through catabolic reactions, they will be broken into smaller, simple molecules. And of course, as a result, energy or ATP will be released. Then simple molecules will go through anabolic reactions. Anabolism means we build up something. So as a result of those anabolic reactions, large molecules will be formed again, and of course energy will be used for those reactions. Before we continue our lecture, let's go through a few terms and make sure that you are familiar with those terms. The first term is biochemical pathways. It is a chain of biochemical reactions, and each reaction is catalyzed by enzymes. Substrate. Substrates, those are compounds that play a role of starting points of reaction. For example, let's look at the bottom of the slide. You see A, B, C, D. Let's say that is a chain of biochemical reactions, or we call them biochemical pathways. For this reaction, A, B, C, D, it's a long reaction, right? A will be a starting point, will be a substrate. What is going to be D in this reaction? D in this reaction will be what we call the end product. How about B and C? B and C are metabolic intermediates. So metabolic intermediates, those are compounds that formed by one reaction of the pathway and used by next reaction. Phosphorylation. Phosphorylation means a phosphate group will be participating in biochemical reaction. Formula of phosphate group is PO4. So when compounds receive a phosphate group as a result of biochemical reaction, we say compounds has been phosphorylated. If as a result of biochemical reaction compounds lose their phosphate group, we say compounds has been dephosphorylated. Enzymes. Enzymes are substrates that catalyze biochemical reactions. So let's go back to catabolic pathways. Remember, there are three catabolic pathways, and altogether they form 12 precursor metabolites. Glycolysis forms 6 precursor metabolites. TCA cycle or tricarboxylic acid cycle forms the next 4 precursor metabolites, and the last 2 will be formed by pentose phosphate pathway. All those 3 catabolic pathways are called central metabolism.